And okay, there we go. Let me just keep the chat open. Okay, and I'll just I'll just make it like Chris Wright style, in the sense that if you have a problem, or if something's going wrong, just just yell out, because I might not be able to reach chat. If I press present like this, can you guys see it? This is working. All right. I apologize, my internet's a bit garbage. Okay, because it's still loading, it's probably not going to work. No patience. Okay, oh, where's it gone? There we go. Oof. All right, hold on. Let me stop sharing my screen, fix this, and then come back in a second. Oh dear. I'm sorry, guys. Probably close the amount of tabs I have. Again, this will be recorded, and you guys can find the slides yourself on in the year 1 2020 folder under revision lectures. So. God. All right, almost there, I promise. I'm so sorry. Okay. I think my drive's dying a bit. Jesus. Okay, I think we're almost there. I seriously apologize for my internet. Oh, at least you guys aren't. Once you're on campus, you'll really love to appreciate Eduroam and how much it can really destroy your internet. Okay, let me try closing a bunch of other stuff I don't need. Okay, I think it should be almost there. I've finally managed to get my presentation open, which is a good sign. So I should just share my screen now, and then we can get started. Okay. All right. My screen should be sharing now. I'm just not gonna click the present button because I think that might ruin my computer a bit. As you can see, it's still loading a bit. But I think this revision lecture has a bunch of content, as I'm sure you guys are aware, microbiome and immunology. They really pack in a lot of content, but as I'll get into a lot of the like yield and stuff about that a bit later, you'll see you don't need to stress about memorizing literally every single detail, because a lot of it will get reinforced in second semester because you have more microbiome there and some more, not too much more, but some more immunology. Hey, come on. Good Lord. Um, okay, yeah, as you guys can see, my computer's not great. Please. Okay, great. Okay. All right, slowly and surely we're getting, I think. I'm really sorry for the late start, but hopefully we can zoom through it. I've got a lot of practice questions at the end that I'll probably skip out on and leave for you guys to look through. So all the content really for today's lecture was taken from last year's revision lectures, Meredith Hughes lecture, the immunology lecture guy, TSP notes. There's a link to my OneNote from last year. It's got all the notes for every single lecture from the beginning of the year to the end of the year. And I think it helped a lot of people out last year as well, or hopefully. Okay, now this is like the relative mark breakdown last year. So for last year for microbiology and immunology, it was only on paper 2 for semester 1. And as you can see, it took out about a third of the paper, both of them combined, and that covered all of this stuff here. I'm not sure what, I hope you guys have had all your immunology lectures already. Someone yell out yes or no, or okay, or not. That's all right too. But I'll still try and cover pretty much everything from both, with exception of a little bit of immunology towards the end regarding cancers and stuff like that, and obviously not pharmacology. Okay, so microbiology, step 1, it's probably Staph aureus, and learn to wash your hands. So we'll just start by talking about microbes. Once my computer starts to go. So all microbes are, as you can see, are classified into 3 main living organisms, and that's based on their rRNA. So there's bacteria, archaea, and eukarya. So basically, you just need to remember that bacteria are prokaryotes, and you guys would probably be drilled into the difference between prokaryotes and eukaryotes a lot, but it's just important to remember that bacteria fall into the prokaryote section. So just a little bit on the differences between prokaryotes and eukaryotes. So prokaryotes are unicellular, simple structure with no membrane-bound organelles or no true nucleus. So, and then they have a slightly more complex cell wall with peptidoglycans and stuff like that. For reproduction, they use binary fission, and they're kind of like extremophiles in the sense that they can survive in a large variety of environments. And the three main shapes that you actually need to pay attention to for bacteria are cocci, bacilli, and spiral. Mostly you'll see cocci, like gram-positive cocci in grape. Like if you hear gram-positive cocci in a grape-like formation, it's Staph aureus. So, and then eukaryotes are basically most other living organisms, so they're union multicellular. A lot of your parasites end up being eukaryotic. They've got a simple cell wall, and they use asexual or sexual reproduction, and they're highly variable. All right, cool. Let's try and zoom through this part here. If my computer wants to work. Oh, no, that's too far. Oh my God.
Hey, Akil.
Hey there.
Hey, if it's easier, one of us can share screen of the presentation while you talk through it.
Yeah, that'd be great, actually, mate. Thank you. Do you mind doing that?
Yeah, I'm just not on the document, but I can get it up. If someone on the document wants to do it, it would be quicker, but I'll get it up as well.
Yeah. Sorry for the shocking internet. I definitely need to. I'll stop sharing my screen then. And if any of you wants to share your screen, probably best to hide any messages beforehand. Just saying. All right.
Okay, so I got it up.
All right. Thank you. Great. Can we go? If you don't mind, could you please go to the bacteria slide? So.
Next one.
Yep. Next one. Just a few more. Yep. The next one, please. Yep. Okay. A little bit about bacteria. Oh, one too far away. Sorry. Yeah. Okay. So bacteria, basically, their asexual reproduction is effectively binary fission. Simple, fast. It's got a three-stage simple process, which is replicating the genome, extending the cell wall and membrane, and then splitting, as you can see from the diagram right there. Regarding the cell walls for bacteria, which you'll probably need to know a bit about, is that it's formed from a peptidoglycan layer, which is a complex carb that can stain purple, which is important for gram staining. And then it's got, for mostly gram-negative bacteria, you have lipopolysaccharides at the top, at the apical membrane. So the part that's kind of the outermost layer, and often that's parts top, like the toxic part, and often toxins are part of that. And then there's also teichoic acid in gram-positive. That's right at the top of the peptidoglycan layer. Not too important though. All right. Yep. Next slide. Thank you. Okay, so regarding gram staining, this will probably have been drilled into you, but there's gram positive and gram negative, and so gram positive is the thick peptidoglycan layer with a purple gram staining, and basically, as you can see, it's the image on the left, which says peptidoglycan plasma cell membrane cytoplasm, and for gram positive bacteria, it's thicker, thus retains more of the stain. For gram negative, that's when they have a thin peptidoglycan layer, and that ends up with a pink gram stain. So your staph and your strep are gram positive, and E. coli and salmonella are gram negative. Also, a lot of your gut bacteria, which is a bit more useful for next year, they end up being gram negative. In some cases, you can't really gram stain because of the differences in cell wall, such as Mycobacterium tuberculosis, which is the one you need to know, and because it has a lipid layer of glycolic acid on top of the peptidoglycan layer, which prevents gram staining effectively. So instead, you use Zeal-Nielsen acid fast staining, which uses different procedure and method in order to elicit a stain. If I'm going too fast at any point, let me know. And you have any questions, again, let me know. All right, next slide. All right, so a bit on the external structures for your bacteria. So as you can see here, they can be divided mostly into by their function, which is protection, motility, persistence, genetic recombination, and adherence. So these are also really important for its virulence factors and how it can actually cause disease. So regarding protection, you firstly have your glycocalyx sugar capsule, which is around the outside of the bacteria. So this is great for resisting phagocytosis and for molecular mimicry. So molecular mimicry is essentially when the bacteria kind of hides itself and prevents immune cells from being able to detect it. It's also very useful for adherence and formation of biofilms. So it allows bacteria to stick together and also can help protect from the harsh environments it may be persisting in, similarly to the slime layer. Regarding motility, you have mostly your flagella, and so that's made from the protein flagellin, and it's kind of used like a propeller. And I'm not sure if you guys have kind of seen like the diagram of, or you probably would have seen the diagram of cilia and stuff for the mucous membranes. It kind of works the same way as that in how it acts like a whip motion and kind of moves things along. Furthermore, for persistence, it's got endospore, so persist in harsh environments. And for more adherence, it has fimbriae or pili, and those are basically short projections from the surface of the bacteria, and they're kind of like flagella, and they kind of really are important in sticking to the mucus layers in your throat. So for when you have cough, often those types of upper respiratory bugs have fimbriae or pili on them in order to stick properly to the cell to the nasopharyngeal passage. Regarding genetic recombination, there are sex pili which are there for plasmid conjugation and genetic recombination. So this is basically when bacteria share genetic information in order to diversify and be able to persist longer. All right, let's move on to the next slide. All right, so what I've tried to do for the majority of this presentation is condense a lot of information into a small amount of slides so that you guys can always refer back to the slides and learn from there, because all microbiology happens to be memorization, sadly. So regarding the host-parasite relationship, which is fairly simple, there's mutualism, commensalism, and parasitism. Mutualism, as implied by the name, is mutually beneficial for both organisms. Commensalism is when a single organism benefits with no expense to the other. So this is the majority of our normal flora are commensals. And then there's parasitism, which is when one organism benefits at the expense of another, which is often pathogenic bacteria or viruses. Actually, not viruses, sorry. Now, next regarding virulence factors, I spoke a bit about adhesion molecules, antiphagocytic factors, and toxins, and those are like the three main virulence factors for bacteria. Then a note about lipopolysaccharide endotoxin is that it can cause septic shock, which is a really important part to remember about the lipopolysaccharide layer. Most of these virulence factors can be demonstrated through Staph aureus, which is why they really like to bring it up a lot, because it's clinically quite relevant, and it has a lot of these virulence factors being a pretty nasty bacteria. And in order to cause disease, for a bacteria must firstly transmit to the host, then enter the host, avoid and overcome the host defenses, grow and spread, and then cause damage to the host. Okay, so we've got so regarding normal flora being considered only commensalism, or can it be also considered mutualism? It definitely can be considered both, but it depends on the specific type of flora. So you have a lot of flora that might live on your skin, for example, like staph epidermidis or something like that, and that's a commensal because it doesn't really do much for you, but like it doesn't do you any harm. But then you have parts of your gut microbiome which are mutualistic, which do exist in mutualism, where it helps excrete. They're important for vitamin absorption and preventing other bacteria slash pathogens from infecting you. So it is both, but the majority of them are commensals. I hope that answers your question. All right. We'll move on to the next slide. Okay, great. Okay, so just now speaking a bit more about the normal flora and the gut microbiome, I have put that photo in there to show you simply the range of and the diversity of the normal flora and the fact that they're everywhere. Please don't bother going and learning all of them because it's not really too useful yet. Like it's too much too soon. You'll like burn out. So it's mostly commensalism, as I mentioned before, and parts of the normal flora are only considered parasitic when they cause infection. So they're opportunistic pathogens. Um, so that's usually when there happens to be a vulnerability to the host, or there needs to be susceptibility and allowed the progression of the disease for the bacteria to be able to enter and infect the host. So you have, like, for example, Staph aureus. You have it on you a lot of the time, and uh, it's only when there is an opportunity or the host is weakened that the bacteria, which might normally be considered a commensal, will become parasitic and become pathogenic to the host. So the gut microbiome and normal flora is acquired shortly during and after birth, but it does continually change. A very interesting point from research is that your gut microbiome actually adjusts to what you eat. So, for example, if you eat a lot of artificial sweeteners, for example, your gut microbiome will adjust to being able to digest artificial sweeteners, and that's why people crave it more often. Well, that and multiple other reasons, but this is why you crave kind of like a lot of stuff that you consistently eat, because your gut microbiome has adjusted specifically to consuming that type of food and used to that the nutrients you provided that way. This is an interesting point, not very relevant. So the gut microbiome is very important for providing protective, structural, and metabolic functions to the host. Those are three main points. And when this goes wrong, you can get gut dysbiosis with a poor diversity within the microbiome, and that can lead to a lot of opportunistic infections. I think Meredith touched a little bit more about it on her lecture, but in her lectures. So if you want a little bit more information, you can check there or in my notes. So I've just put there like roughly for skin, normal flora is mostly gram positive. In the GIT, most of the normal flora there are acid tolerant, and a lot of the bugs in there, or pathogenic bugs, often happen to be gram negative in the GIT. In the genitourinary tract, there is bacteria from from the skin, the colon, and the vagina, and I believe the upper urinary tract is meant to be sterile. So the upper respiratory tract has both gram positive and negative normal flora, and there is no normal flora normally found in the lower respiratory tract or upper urinary tract. Therefore, any bacteria which is found there is pathogenic or not meant to be there. All right, let's move on. Speed up.
So I think there's a bit of a lag from when I change the slide to when you see it.
Oh, no, that's okay. All right. Here is a bunch of important bacteria. I'll just name a few, and you don't worry, you don't need to memorize all of these because you'll actually die. Like, there's no point memorizing all of these at once. So a few ones that are important are Clostridium perfringens, which causes gas gangrene. You have your MRSA, which is your Staph aureus. Then you have, for tuberculosis, you have Mycobacterium tuberculosis, strep throat for strep pyogenes. Then for your fecal oral route contamination, so you've got cholera, which is a big one. Then you've got E. coli and salmonella. Clostridium difficile is pretty important for nosocomial infections. The rest of it, pretty much you can get away without knowing. Maybe you probably need to know meningitis and Helicobacter pylori. Yeah, I pretty much listed them all, but don't worry too much about it. Okay, let's move on to the next slide. All right, so a little bit about viruses. I'll zoom through this. So buzzwords here: acellular, obligate, intracellular parasite, which is a fancy way of saying it can't live on its own. It's essentially like a 4-year-old kid. Like it needs a parent. Like it needs a host to live, otherwise it can't do anything. So it generally contains genetic information that can interfere with normal cell function, and each virion, which is a virus particle, contains a nucleic acid, a capsid, and sometimes a lipid envelope, which is formed from the host cell. So that's, for example, in HIV after it infects a T cell. Then the classification is generally based on nucleic acid. And each virus usually has one of them, has either a single-stranded RNA or blah, blah, blah. Regarding, then it's also classified based on its morphology, so it's icosahedral or helical, then whether it's enveloped or not, and the hosts it infects and the range of hosts it infects. So it can be a narrow range type of virus or a broad range. So a broad range would infect humans, animals, etc., as well as the disease produced. A little note on retroviruses. So retroviruses are single-stranded RNA viruses, and they effectively use the reverse transcriptase mechanism, which I believe you guys cover in an ICL about HIV. And that's effectively how they integrate their genetic information into the host DNA. So for HIV, that basically happens because of the formation of complementary DNA from once the HIV particle enters the cell, and then through the enzymes of integrase, it fuses with host cell DNA, and then it can reduce virus particles or remain latent. Okay, next slide, please. So viral replication, there are 5 main stages. It's pretty intuitive in the sense that if you think of it and break it down in your head, this is the steps that it has to undergo. So it has to attach to the cell firstly, then it has to enter the cell, then while it's inside the cell, it needs to replicate to produce more viral particles. Then those viral particles, while within the cell, capsids form around them under the assembly phase, and then they get released. Yeah. All right. Next slide, please. So the viral disease progression. So the 8 stages on the left here are the viral disease progression, which is similar to the replication to some extent, except that you can kind of be split into 2. So you've got firstly your entry, then your primary replication, which is your site of initial infection. Then you have the innate immune response, which will occur. And if the virus survives, then it can be incubated and stay within cells. So, for example, HPV, HIV, and then there might be a secondary replication which occurs. So, for example, varicella zoster secondarily replicating into shingles and stuff like that. And then you have the host immune responses, then release transmission, and then you can either get the resolution of infection or chronic infection. They touch on the outcomes of virus infections regarding hepatitis. So be sure to watch out for that one in your active learning on it. Then a note on influenza, because I like to ask questions about influenza. So basically you need to know about hemagglutinin and neuraminidase, big words for receptors effectively. So hemagglutinin is essentially the entry / attachment receptor, and then for release is neuraminidase. So example, neuraminidase inhibitor seltamivir prevents the exit of influenza from the cell, which is how it works. That's just a little pharmacology plug. So regarding the types, you only basically need to know that know about influenza type A. So that's the most regular for outbreaks, and that undergoes antigenic shift and drift. And I believe influenza B can also undergo antigenic shift. So antigenic shift is the big bad one. So you can just memorize it. Like the way I memorized it was like antigenic drift is a slight change, and antigenic shift, I just forget the F and it's antigenic shit because it's horrible. Like that's not what you want to see. So antigenic shift is the mixing of 2 strains within one host cell. So that's 2 strains. So that's when they've a single host cell has been infected by two types of influenza, for example, and then the two strains mix while integrating, and then you get a whole new strain which you have no natural immunity to, and these are like the cause of your major pandemics, such as like the avian flu. Antigenic drift is mostly for seasonal flu, and there are point mutations in either the hemagglutinin or neuraminidase receptors. Yeah, next slide. Let me know if I'm going too fast, or if you want me to speed up. So here are some important viruses. Honestly, do your best to like learn them. Rotavirus, norovirus is for diarrhea and gastro. Do keep in mind, if you don't need to stress too much about memorizing them this very day. You'll eventually get through and learn them. And I've put in the brackets there some important parts about it. So like the papillomavirus is oncogenic, similarly with Epstein-Barr virus causing Burkitt's lymphoma. Yeah, okay, let's move on there. All right, so a little bit about fungi. I'll be honest, they don't really ask me any questions about fungi, but I guess they're important to know, and they're only really like talked about in cases where the patient is immunocompromised, or people with HIV. Like you don't, like most normal people don't get fungal infections, with the exception of maybe like some tinea or like athlete's foot or something like that. But like systemic fungal infections are almost exclusively reserved for like people who are exceptionally immunodeficient. So that's a very like big sign of AIDS, HIV turning to AIDS, is when you have people with fungal infections, or like systemic fungal infections. Oh, so characteristics about fungi is that they're eukaryotic obligate aerobes, which means they need air to survive, which is important. So that's why the location of the infection often requires air. Instead, relative to other like bacteria and viruses, is the cell wall is made of chitin, and the important point is that within the cell, it uses ergosterol instead of cholesterol, as where we use cholesterol in our cell membranes, and this is often a pretty big target for most of your antifungal agents. So there are three types. So you've got yeasts, molds, and dimorphic fungi. For some reason, they ask about like the difference, like for dimorphic fungi, like what temperature it might be a mold, what temperature it might be a yeast. This is clinically relevant for people with fever. So if it changed from a mold to a yeast at 37 degrees, we got fever. It's kind of important. And so yeasts effectively grow via budding, which is a form of asexual reproduction, whereas molds grow by extending hyphae, which is big long strains. It kind of looks like a spider. So regarding allergies, it's generally due to inhalation of spores or cells from the mycelium. Fungal toxicosis is effectively when mycotoxins, which are small metabolic products, which are there in badly stored food, they cause poisoning, and that can cause respiratory distress, burning sensation, swollen lymph nodes. And the severity of your fungal toxicosis depends on the type of fungal mycotoxin that's being ingested, the amount, and obviously how long you've been exposed to it. All right, let's go to this one. Okay, so here are like the more important fungal diseases. So you got your tinnias, which are like your cutaneous fungal diseases. So you got tinea pedis, athlete's foot, and interestingly enough, ringworm, not a worm, as they would have told you, it's a fungal disease. Subcutaneous ones are very difficult to treat. Then you got your opportunistic and invasive ones, which are the ones that you see in immunocompromised patients, such as those with HIV. So candida, so that's like your that's like your thrush. Then you have aspergillosis, so that's from your aspergillus fungi. And then if you have fungal pneumonia, that's like it's pretty much guaranteed that you've got AIDS at that point, because your immune system would be really, really bad for you to be able to contract fungal pneumonia from that specific bug. All right, let's move on. Okay, quick note on parasites. So these are your helminths, protozoa, and ectoparasites. So your helminths are your multicellular eukaryotes. So you got like three main types, which are nematodes, cestodes, and trematodes. Nematodes are most common, so those are roundworms. Cestodes are tapeworms, and trematodes are your flukes. So they can range from being as like a few centimeters to like meters long. And their most common form of transmission is from fecal oral ingestion. So this is why kids get a lot, because kids are kind of nasty and they don't really wash their hands a lot, and they put their hands where they really shouldn't. So this is why you get kids with like well, let's speak about the specific bug, like specific helminth later, but this is why a lot of kids end up getting parasitic infection with helminths. Protozoa are unicellular eukaryotes, and those are transmitted by vectors, like a vectors, maybe like insects, or ingesting the infective agent or the eggs or parasite. Ectoparasites, they don't cause disease, but they can transmit them. So they're basically arthropods. They're not really asked about too much. Just if you hear ectoparasites, basically they don't cause disease. All right, see. Let's move on to the next one. So these are the actual this is the part of parasites you might need to know. If you remember the lecture, the guy chucked up a big slide with a lot of non-English words. I remember that was a good source of PTSD from year one. So you've got, for your helminths, so you've got hookworms, and those live in your small bowel and cause iron deficient anemia. Enterobius was the worm I was speaking about before, which is a pinworm, and that's common in children. And basically, they live in the cecum, where at night they crawl out of the anus at night to lay eggs. This is very common in children, and this is how when kids end up sick, and when their parents usually go clean their diaper, they sometimes at night they can see this, and then they freak out, and they're like, "Oh, okay, we need to go take it to hospital," and then they'll just treat the infection. Filariasis, a buzzword, can lead to lymphedema, and that's from the black fly. Elephantiasis is effectively a far more severe form of lymphedema, where you've got massive swollen like legs and calves and stuff. And then a little bit about African sleeping sickness there from the Trypanosoma species. Next, regarding important protozoa, so malaria is actually important. Again, the lecturer would have drilled to you that if there's fever and return traveler, always suspect malaria. So that's from your Plasmodium species. So malaria infects the blood and the CNS. So this is clinically relevant for the blood, because obviously if you find malaria in the blood, it's bad. And this is also important for people who might have had splenectomies, so people who had their spleen removed, as we'll talk a little bit about later. The spleen's kind of like the lymph node for the blood. So if you have no spleen, your malaria infection can be really, really bad. Then the five main species, or the five main Plasmodium species that transmit malaria are written there. The one you actually need to know is Plasmodium falciparum. That's the one that's the most severe and the most common. And then just kind of have a vague memory of knowing which ones of these actually are malaria, because they might chuck in as a question, which one of these cause malaria, and it'll be a list of 4 of them and 1 extra that you've never heard of. So it's transmitted by female Anopheles mosquitoes. That's something that they want you to know. And again, interestingly enough, hydroxychloroquine, which Trump is using as a prophylactic for COVID, is an anti-malarial drug. So yeah, the main point for malaria is to prevent it. So a lot of people who travel can take prophylaxis. There are malaria nets, use mosquito repellents, and the best way of preventing malaria, especially in developing countries, is by providing like support through donations of nets and stuff like that. Next, you have Giardia, which is mostly from drinking bad water. So that causes, like, a buzzword is watery then greasy stool, and you treat it with metronidazole. Toxoplasmosis gondii, like the way I just remember it is that it's from like eating kitty litter, effectively. It's mostly a self-limiting disease where it causes flu symptoms, but it's important for immunodeficient individuals because it can cause cerebral lesions then, and that's pretty grim, as implied. All right, let's move on to the next one. Okay, so terminology and transmission. So here I've got, on the right, I've got like a diagram of transmissions and the types of transmissions. So you got respiratory, fecal oral, venereal. Then you have regarding arthropod-borne infections and zoonosis, you've got like through through a vector such as like mosquitoes, or like a vertebrate reservoir such as like a dog, or a vector vertebrate reservoir. So the reservoir obviously kind of just means where the disease actually stays. Then I've got a few definitions on the left there. So you've got epidemic, endemic, outbreak. These are some like more common words. Hopefully you guys recognize them. Just kind of memorize the definitions of those ones, or like just roughly know them. All right, let's move on. I'm sorry, I'm trying to move a bit quickly, and I think I'm a bit late. Okay, so this is a chain of infection. Basically, in order for in order for an infection to occur, this has to happen. This is just like the pathway that goes through. All right, let's go on to the next one. Okay, regarding the control of microbes, this is like a largely common sense, but they want to teach it because it is really crucial in nosocomial infections, and problems with this is a very large reason why nosocomial infections occur. So within a healthcare setting, we have like the three main things of cleaning, disinfecting, and sterilizing. So cleaning is just the stuff that you wash with like hot water and detergent, and that's like tissue, linen, dressing, stuff like that. Disinfecting is removing or killing most of the viable organisms, so that can be done physically, such as like boiling water, or like chemically with things like chlorhexidine and stuff like that. And then you have sterilization, which is used for anything that can penetrate your mucus or skin. So that's like removing all viable organisms, and that's really important to prevent sepsis. So for these are like the basic rules: is that anything that enters the skin, sterilize it. So like a catheter, anything that may contact skin but not penetrate it, so like an X-ray equipment or like linen, just disinfect or clean. And then human hands, disinfect your hands, use chlorhexidine. Don't mess up your your OCEs because you don't wash your hands. And then some other equipment requires some specialized decontamination. Preventing the growth of microbes is pretty important by preventing any access from microbes from just being able to proliferate, such as keeping it at a specific temperature, specific moisture. So in a lot of moist and warm environments is where bacteria like to proliferate. So yeah. All right, let's move on to the next one. Okay, so these are methods for disinfecting. These, like, it's kind of annoying, but you like kind of need to, they want you to like vaguely memorize for at least disinfecting. Like those are the three main ones. Let's go to the next slide. The next one for sterilizing, they want you to memorize. They will almost like 100% ask you a question on like dry heat, moist heat, and pressure, and like the temperatures. So just remember hot air oven regarding dry heat, which is 170 degrees Celsius for an hour or 160 degrees for 2 hours. And when they use moist heat and pressure, it's through an autoclave, and that's 134 degrees Celsius for 3.5 minutes or 121 degrees Celsius for 15 minutes. So just remember those two. Yeah? All right, cool, yeah. Let's move on to the next slide. Okay, nosocomial infections. As I said, it occurs when there's a breakdown in sterilization or disinfection procedures. So follow your cleaning protocol and stuff like that. And I've put on the right the most common types of nosocomial infections and where they happen. So the most common type of nosocomial infection are in the urinary tract, and that's because catheterization and sterilization from there is very common. And so if you see any of the ones on the bottom right, like Staph aureus, Klebsiella pneumonia, E. coli, Clostridium difficile, Pseudomonas, those are like very, very important nosocomial infections. And nosocomial infections often end up being really, really bad. Like they're very severe. So your methicillin-resistant Staph aureus, nosocomial infection. A lot of those patients end up being on antibiotics and other drugs. So for them to get an infection that proliferates often needs to be quite a severe one. Yeah, let's move on. Excellent. We're done with microbiology. So, okay. I'll probably take another half an hour or so on this. It's all right because I told the other lecturer he'll start at about 11:20 for the next one. All right. Let's go to the next slide. Okay, so let's just broadly break down the immune system. So the immune system consists of innate and adaptive immunity. And so your innate immunity is your initial defense, and it's got no lasting memory, and it's immediate onset. So this is like immediately what happens to prevent microbes from causing disease. So then you have your adaptive immunity, which is quite a specific immune response to eliminate pathogens, and this is quite long lasting, and it can create a memory T cells and stuff like that for faster and more vigorous response to individual pathogens. And that can last from like hours to days, depending on the infection. Yeah. Let's move on. All right, a little bit about lymphoid tissue. So you got your primary and secondary lymphoid organs. So primary are where your immune cells are made, so like the bone marrow, the thymus, and your secondary lymphoid organs, such as like your lymph nodes and your spleen. So the lymphatics are basically system drainage and allows for detection of antigens in tissues. And the lymphatics system runs through lymph nodes, which can be considered surveillance centers. So mostly like a little known them. So the antigens on antigen presenting cells, which we'll get more into later. So they enter the lymph node via afferent lymphatic vessels. For those of you who end up getting like a little bit confused between afferent and efferent, the way I remember it is afferent as in affect, so before, and efferent as an effect, so after. Then T cells and B cells enter through the arteries via the high endothelial venules, and then after activation by the antigen presenting cells, they leave the cell. So this is basically where like antigen presentation occurs within regional lymph nodes, where the antigen presenting cell presents itself to T cells, B cells, and then those guys exit. And we'll cover more about that later. And then spleen detects bloodborne antigens, and again, is kind of like the lymph node of the the lymph node of the blood system. All right, cool. Let's move on a bit. All right. So the cells of the immune system. So these cells, you have myeloid and lymphoid cells, and these originate from hematopoietic stem cells, which are from the bone marrow. So you just kind of need to like know which ones are myeloid and lymphoid. For now, it doesn't matter too much, but like they'll probably ask you a question, which one of these from myeloid lineage or lymphoid lineage. And a good thing to know is that mostly your your lymphoid cells are your adaptive immunity, and for your myeloid cells are often innate immune cells. Yeah. Let's go on. All right, so a little bit about the innate immune system. So the first line of the innate immune system is your natural barriers. So your anatomical barriers, such as skin, mucous membranes, coughing, sneezing, like vomiting, stuff like that, which tries to effectively remove the pathogen from your body without any like real immune activation. Some of the antimicrobial and chemical barriers are like lactic acid in the skin, low pH in the stomach, lysozyme in tears, sweat, saliva, and serum. And then the second line is where it gets a little bit more complex. So the second line of the innate immune system. So you've got your complement activation, inflammation via the inflammasome, your PAMPs, your PRRs, and then a bit about your white blood cells. All right, let's move on. All right, so complement. As you can see, big slide. A lot of people panicked a lot about the complement because Guy kind of ran over it pretty quickly. It's not like it's vaguely important to just understand what it does and kind of know the different C3, C3b, stuff like that, to just know like which one. I'll show that in the next slide for the mechanism. But it's important to understand that the complement system is a mechanism of the innate immune system that's always present in the serum, and it's effectively like a chain of mechanisms and reactions which involves a bunch of cleaving that effectively tries to destroy and kill bacteria. It's a very like common mechanism for trying to kill bacteria and pathogens. So you got your three pathways. You got your lectin, classical, and your alternative. And then that ends up with three outcomes of recruiting more immune cells. So that's like inflammation, increasing phagocytosis, and then the formation of pores in the pathogen surfaces. So that formation of membrane attack complexes, as you can see in the bottom right there. So why do we actually use a complement system? One is for opsonization. So that's when the proteins bind to the pathogen and kind of act like a big beacon to the phagocyte. So that's how it increases phagocytosis through opsonization. Chemotaxis, as implied by the name. So chemochemical, taxis as in, taxi. That's like that's encouraging the movement of macrophages and neutrophils to this encouraging the movement of macrophages and neutrophils to the cell, which has been infected via chemokines and cytokines. Then the next is creating cell lysis via membrane attack complexes, which are formed. And those ones basically punch holes and kill the bacteria through that way by essentially all the fluid running out once you punch a hole in it. Then there's also agglutination, which is when antibodies cluster pathogens together to make it easy for mass phagocytosis. I know I've gone through that bit a little bit quickly, so stop and ask me any questions. I've got another slide on it, so let's go to the next slide, actually. Okay, so this is complement round 2. So a little bit more on the classical pathway. So the classical pathway is essentially when the molecules bind with the pathogen surface or with the antibodies bound to the surface of the pathogen / cell. And so that's when that encourages phagocytes with receptors to engulf and destroy the pathogen. The lectin pathway is through carbohydrates, which are bound to the surface of the pathogen, and they effectively recruit phagocytic cells to the site of the infection and promote inflammation. So that's the inflammation and chemotaxis aspect of the complement system. Then the alternative pathway, which is the amplification pathway, and this is what leads to the formation of membrane attack complexes, and that's what disrupts the cell membranes and actually kills the cell. As you can see on the right here, you've got like the 3 main pathways, and the boxes over there are like the kind of important things. So so you have C3, and then after you have C3 in the cells, and then through the initiation of complement, that cleaves the C3 into C3A and C3B. And C3A is what causes inflammation and effectively promotes a bunch of interleukins or like a cytokine release and causes cells to come towards that specific cell. Then you have C3 for opsonization and phagocytosis, which is deposited on the microbe. C5A causes further inflammation, and C5B, which attracts C6 through 9, forms the membrane attack complexes. I've written the mechanism right there in a little bit more detail. So those are like, I apologize, but like those are like the things you kind of need to know. So like C3, cleaving to C3A, C3B, C5, cleaving to C5A, C5B. The C3A and C5A via the classical pathway promote inflammation. Then the C3B binds to the pathogen to attract phagocytes, which is the lectin pathway. And then the bacteria can't be phagocytosed until C5A activates some macrophages. And if you see on the right there, C5A is used for inflammation. So once the macrophages arrive, then it can get activated through C5A. And then C5B attracts C6 through 9 and forms the membrane attack complex. And that's the end of the alternative pathway. That's a bit complex and it's a bit tricky. So maybe a good idea is to read through this maybe a couple times or rewatch that part of the lecture. Yeah. All right. Let's move on. Okay, inflammation and the inflammasome. So the aim of it, as soon as people hear inflammation, they automatically think it's bad, but inflammation is actually a good thing. But excess inflammation and chronic inflammation is bad. So the aim of inflammation is to eliminate pathogenic insult, to remove injured tissue components, and to restore homeostasis. So the 5 cardinal signs of inflammation that they like you to know is calor, rubor, dolor, tumor, functiolacia. So that'll get drilled into you enough, so hopefully you remember that. So inflammation can be triggered by cell damage and not just infections. So that's the cell damage promotes inflammation through damage associated in molecular patterns. And the inflammasome is effectively a structure within cells that effectively promotes acute inflammation. So the process is like a lot more complex than you really need to know. But as you can see in this photo, you start off with NLRP3, which is kind of like a sensor. and that responds to bacterial products. And what activities they do. So whenever potassium leaves a cell in like a dangerous manner, then NLRP3 is stimulated by that, and that binds with an adapter and caspase 1, which is an inactive part, and those three combined. So the sensor, the adapter, and the caspase, they form the inflammasome, and effectively through the mechanism of pathogen associated molecular patterns, so PAMPs, they activate the synthesis of inactive cytokine pro-IL-1 beta. So as you can see in the bottom like left of the photo there, so that's the innate signal. So stuff like TLR3, which is an intracellular toll-like receptor, that through the innate signals that produces pro-IL-1 beta, and through the combination of active caspase 1 and pro-IL-1 beta, you get the formation of interleukin 1 beta. TLDR inflammation, or like the inflammasome, generates interleukin 1, which is pro-inflammatory. All right, cool, let's move on. Regarding pathogen-associated molecular patterns and pattern recognition receptors, so PAMPs are structures unique to pathogens that are recognized by the innate immune systems. And these are things that the pathogens can't really evolve well from. So these include like surface structures such as lipopolysaccharides on the gram-negative bacteria on the outer cell walls, flagella, double-stranded RNA, sugar residues, and unmethylated CpG nucleotides. So pattern recognition receptors such as your toll-like receptors are what detect your PAMPs, and binding to different receptors will result in a different cascade of immune event. So your toll-like receptors on your extracellular membrane are your TLR4 and TLR5. So TLR4 is for your lipopolysaccharide recognition, and TLR5 is for flagellin recognition. Then on the endosomal membrane, so inside the cell, as I mentioned before, you have toll-like receptor 3, and those recognize viruses through TL3 recognizing dsRNA. Then you have some other receptors in there. They're not too important. Let's move on. Okay, so your interaction between toll-like receptor and PAMP, you'll cover this as well a little bit more next semester, but I'll just mention it for now. So effectively, the interaction between toll-like receptor on the immune cells and the pathogen-associated molecular pattern initiates phagocytosis. So phagocytosis can be undergone by macrophages, monocytes, dendritic cells, and neutrophils. And so phagocytes essentially is the internalization of pathogens and then release of mediators to break down the pathogen. So first you have the ingestion, you have the pathogens which are engulfed by a phagocytic cell through endocytosis. They get chucked into a vacuole called a phagosome, and that binds with a lysosome which is within the cell, and you get the formation of a phagolysosome. And within the phagolysosome, you have lysozymes, so enzymes and free radicals and hydrogen peroxide, and they effectively break down the pathogen. You'll learn a little bit more about in bacterial pathogenesis next year how some bacteria can effectively avoid this or survive through it. All right, cool. Let's move on. Okay, a bit about cytokines. So cytokines are just small soluble molecules secreted by cells. And chemokines are effectively a type of cytokine. So the roles of cytokines is like inflammation, coagulation, and regarding inflammation, this has to do with, they would go through the whole like rolling and sticking with the adhesins and stuff like that within blood cells. So cytokines allow the blood vessels to become more permeable through tumor necrosis factors, so TNF and IL-1. And then they kind of bind to proteoglycan and express the increased expression of surface adhesion molecules, which allows for the whole stick and roll thing. Then you have like a little bit on type 1 interferons. I'm not too sure if you guys covered it yet, but you have interferon alpha and beta, which are released by virally infected cells. Then the effects of the interferon is that they activate natural killer cells and other immune cells to kill the infected cells. And they also bind to receptors on neighboring uninfected cells to try and activate more signaling pathways to prevent viral replication and kind of call more NK cells and stuff like that. All right, cool, let's move on. So a bit about neutrophils. They're the most abundant. They're not always found in tissues, and they're characterized by their lobulated nuclei, as you can see by the photo there. The production of neutrophils ramps up a ton with infection, and they're kind of like rapid action and passes effectively a bunch of dead neutrophils. So the way that they work is through phagocytosis. So they either phagocytose pathogens on encountering them, or through netosis. I remember getting asked a question on that last year about netosis. So effectively, that's like it ejects a net of DNA histones and antimicrobial agents to kill pathogens. Interesting point: Staph aureus has enzymes to effectively avoid netosis. Yeah, let's move on. A bit about the macrophage. So a macrophage, when it's in the bloodstream, is a monocyte, and then when it's in tissue, it's a macrophage. They're effectively the same thing otherwise. So they're kind of like free, and they wander between cells, and then if they see any pathogen, they'll kind of just engulf them. So they'll signal immune cells about pathogens, and they produce cytokines such as TNF and IL-1, which are pro-inflammatory cytokines, and then they phagocytose pathogens as well. All right, cool. Let's move on. Natural killer cells. So this is what I was talking about with the interferons. So natural killer cells are very useful against intracellular pathogens such as viruses, and they're like the only innate immune cells that is lymphocyte, in the sense that it like recognizes infected and stressed cells and responds to them by killing them or secreting the macrophage activating cytokine interferon gamma. So if the cell is healthy, a natural killer cell binds to the MHC1 on that cell. So every single cell has MHC1, and that's basically like something that's within the list telling them I'm okay or not okay. So usually if it's normal, the NK cell will bind to the MHC1, see that it's activated and that the MHC1 is getting expressed, and it will not kill the cell. But then if the NK cell binds to the MHC1 and there isn't an expression of MHC1, then it's like a sign that the cell has been virally infected, and the NK cell will then kill the cell. Yeah. Okay, cool. Next slide. Okay, I'll do this one very quickly. So these are mostly mast cells, histamine, eosinophils. Eosinophils are for like parasites like helminths and stuff like that, and they release granules with proteases and reactive oxygen intermediates, and that are basically used to kill helminths. So when you think eosinophilia, which is a high amount of eosinophils, it's probably because there's a parasite like a helminth. Mast cells effectively release histamine, and histamine is good for vasodilation, wound healing, and this is like allergies and stuff like that. You'll cover that a little bit more next semester. All right, let's move on to the next slide. Moving on to the adaptive immune response. So, okay, I've got another about 10 or so minutes. Hopefully I can get through it. So this is basically when it recognizes and remembers specific antigens. So it's extremely specific. It's got specialized responses such as humoral and cell-mediated adaptive immune responses. And the response to pathogen improves with repeat exposure. So there are just some words on the right there that you should probably know. So an antigen is a substance that triggers an immune response. So that's on the pathogen. An antigen-presenting cell is the cell that engulfs the pathogens and displays them with their markers, so their MHC2 markers to activate other lymphocytes. Antibodies, which not to get mixed up with antigens, these are like your IgGs, and they're basically produced by B cells. So they're effectively a B cell receptor. And the epitope is the part of an antigen that binds to a lymphocyte. All right, cool. A little bit about lymphocyte development, not too important. Both originate from bone marrow, but B cells mature in the bone and T cells mature in the thymus. And they basically undergo a screening process to kill a bunch of defective cells. And once they're matured, they'll circulate in the blood or lymph to reach peripheral lymphoid tissue. All right, cool. Let's go to the next one. So this is this isn't ridiculously important, but it is kind of important to get your head around it, because problems in this stage in regards to the natural selection of B and T cell development is what leads to allergies and autoimmunity. So effectively, you have your progenitor cells, and then you have your pro or like your pre B and T cells, and the main thing to know is what I've circled there, which is that you want your Bs and T cells to have a weak antigen recognition, and then they'll get positively selected and continue to proliferate. You don't want strong antigen recognition because that's what can lead to autoimmunity and allergies, because you get too extreme of a response and a hyperactive T cell, effectively. Yep. Okay, cool. Let's move to the next slide. Dendritic cells. So dendritic cells effectively process and present pathogens. Their function is like phagocytosis and being the main antigen presenting cell, and they take up pathogens in the periphery and then migrate to the draining lymph node and activate your naive T cells. So they're your main APCs. As you can see on the left there, that's like a pretty great diagram of what you need to know about dendritic cells and how they work. So learn that diagram. All right, let's move on to the next one. All right, T lymphocytes and T cell receptors. So T lymphocytes mature in the thymus, and they travel in blood, and the naive T cells travel in blood and lymph. So these ones are activated by dendritic cells, and you either have CD4 T helper cells or CD8 cytotoxic cells. So this is the rule of 8, where you have CD4 with MHC2 and CD8 with MHC1. This is kind of like something that you need to get your head around, because this means that CD8 cytotoxic cells bind to MHC1, which is expressed on basically every single one of the cells. And so this is why you get more you get a lot more cells which are killed, whereas CD4 T helper cells are for more MHC2. So that's for like longer lasting and B cells and stuff like that. So the functions are to see the antigens via T cell receptors, and each T cell has a unique receptor specificity in the binding site region. And the T cell receptors binds to the MHC peptide combination on the APC, which means that they bind to process bits of pathogens and not to pathogens as they normally appear. So this is why like your CD8 cytotoxic cell is not like, it's not the same as a macrophage or a neutrophil in that sense. Like it's not just going to see a pathogen and kill it. It needs to have presentation to it before it can then kill it. So a TCR or a T cell receptor contains two asymmetrical chains, an alpha and a beta, with variable and constant regions. So the variable region, as implied by the name, is what gives the T cell its specificity, and the antigen binding site is different on each chain. The whole variability point is pretty important, because this is how you get such a massive array of T cells, and basically the formation of T cells and responding to different bugs and pathogens is through mass production and just having so many different combinations that one of them will bind to a specific type of pathogen antigen. Yep. Okay, cool. Let's go to the next one. So B cells. So B cells and antibodies, so they mature in bone marrow and they see antigens via B cell receptors, which is effectively an antibody. Antibodies bind to pathogens as they appear in nature in the normal form, and they're Y-shaped. So you've got two identical heavy chains and two light chains, as you can see in the photo on the right. And each chain has a constant and variable region, and within the variable region, each chain has three complementary determining regions, which allow for the high specificity of antibodies. So CDRs. Yeah. All right, cool. Let's move on. So these are the isotypes of antibodies. So an isotype is kind of different types of antibodies. And so you got your IgA, IgD, IgE, IgG, and IgM. So IgG crosses the placenta, which is important. IgA is for mucosal immunity. So that's a lot of like a lot of your GIT and stuff like that has a bunch of IgA. You got IgG, which I already mentioned IgG, and the IgE is for allergies and helminths. So that's a good thing to know. All right, cool. Let's move to the next one. Okay, antibody and T cell receptor diversity. So this is a bit complex and confusing, but this is essentially for allowing a massive combination of antibodies and T cell receptors. So you got variable regions, and within them, those are formed from DNA recombination, which has a combination of V, D, and J segments. And each light chain contains a single V and a single J, and each heavy chain has a V, D, and J, and the shuffling is random. All right, cool. Next. So a bit about MHC. So MHC class 1 is present on all nucleated cells, and it presents antigens for viruses, and that's on the inside, and that activates CD8. MHC2 is present only on dendritic cells, macrophages, and B cells. So anything with antibody responses is only as a result of MHC class 2 presentation, really. Then you've got, yeah, okay, cool. Let's move on. All right, so this is the general phases of your adaptive immune response. So I'll quickly delve into each of the different parts. So you've got antigen recognition, lymphocyte activation, antigen elimination, and then memory. Yeah, let's move on. Firstly, to antigen recognition. So antigen recognition and presentation. This is like, I remember seeing this diagram and thinking, well, what's going on? It's because it's a bit crazy. So you've got your antigen presenting cell, which processes and presents the pathogen peptides in MHC class 1 and class 2 receptors. And so this is like a dendritic cell which travels from the site of infection through lymphatics into a local lymph node and effectively finds the right T cells from all T cells available. And then the complementing CD8 T cell and CD4 T helper cell bind to this APC, MHC class 1 and class 2 receptor, and then get activated from there. So that's like if you see on the top right or like the upper half of that diagram, you have the class 1 MHC molecule, which binds to the CD8 cytotoxic T cell. Yep. Then regarding class 2, you have class 2 binding with an accessory protein to activate your CD4 T helper cell. So B cells also process and present pathogens to CD4 T helper cells on the MHC class 2 receptors, and through the process of co-stimulation is through the process of co-stimulation activates the B cell. Where is the? I've got a question. Where is the peptide in this diagram? Okay, so when it says processes and presents pathogen peptides in MHC class 1 and class 2 receptors, so that's on the that's kind of on the inside of that cell on the top left. So that's the antigen presenting cell. So the pathogen peptides are kind of like within it. So like the antigen fragments and stuff like that. So peptide and antigen are synonymous in the sense. Okay, cool. Let's move on to the next one. Move on to the next stage, which is lymphocyte activation. Yep, next slide. Okay, so lymphocyte activation is effectively when T and B cells have been activated, they rapidly proliferate in the form of clonal expansion, as you can see right there. So this is basically once they have found the antigen presenting cell has found the correct T and B cell to be able to kill this pathogen, they get activated and then they mass produce, like they go crazy and create a bunch of B and T cells, which create the cell-mediated response for the CD4 and T helper cell, the CD4 and CD8 T cells. So they are the cell-mediated response, and effector B cells, which create the humoral response by secreting antibodies. And there's also the production of memory cells. So important part is just there's first an antigen recognition, which means that the body has recognized the pathogen, and now it finds the T cells and B cells which can respond to that pathogen. And once it finds that activated T and B cells, they proliferate massively in form of clonal expansion, and then they do like antibody secretion, isotype switching, maturation, and create memory cells. Yeah. Right, next. All right, next slide again. We're showing again, and now antigen elimination. So this is effectively killing the pathogen. So CD8 cytotoxic T cells, once they're activated, they can recognize antigen peptides on MHC class 1 receptors. So once they've recognized a cell, the CD8 cell can trigger destruction of the cell by injecting granules of perforin or granzymes. So these trigger apoptosis and effectively punch holes in the cell membrane to kill the virally infected cell. So regarding the cytokines in CD4 T helper cells, so you got IFL gamma and IL-12. So these stimulate CD4 cells to form T helper 1 cells, and these stimulate macrophages, B cells, to fight intracellular pathogens. So this is through the mechanism of opsonization. Then next you have interleukin-4, which stimulates CD4 cells to form T helper 2 cells to stimulate eosinophils, and B cells to fight helminths and allergens. There's a diagram on the next slide, which might make a little bit more sense. The T helper 1 and T helper 2 cells isn't too important right now, but this year you'll learn a little bit more about how the role of T helper 1 and T helper 2 cells in allergies and problems with that. Regarding antibodies, so what antibodies can do is either neutralize, opsonize, or activate complement. So neutralizes effectively binding to the bacterial toxin and prevents interaction with the host cell. So the virus is useless now. Opsonization is when it coats it and acts kind of like a beacon for phagocytosis. And then you have activation of complement, as we mentioned before, which is through killing the pathogen via membrane attack complexes or opsonizing the pathogen. So, all right, cool. Let's move on to the next slide. We're getting close to the end, I promise. So these are basically the diagrams that are very helpful for the adaptive immune response and what the different cells do. So you've got your CD8 cells and how they kill the pathogen on the left. You've got how your T helper 1 cells effectively work, which is your interferon gamma and interleukin 12. And then you have your T helpers 2 cells on the right, which is through the mechanisms of interleukin 4 and interleukin 5 for eosinophil activation and stuff like allergies and stuff like and helminths. Okay, so T helper 1 cells intracellular pathogens, and then T helper 2 cells more for extracellular pathogens. All right, cool. Let's move on to the next one. All right, cool. Well, next part is memory. Cool. Let's move on to the next slide. We're very close to the end, I promise. So memory and acquired immunity, this is just kind of, the diagram on the left is kind of important to show you how you have your initial immune response, which is like your immunization. So you have your first immune response, which goes up and goes down as the infection progresses. Then you have like a booster, which is a slightly quicker immune response. And then because of the memory, like 3 years later, if you get the same pathogen, you'll get a single spike because of your immunological memory and the formation of memory B and T cells as they've responded to that pathogen before. Then that little table on the right, it's a good idea to memorize that because they'll ask you about like, is a vaccine artificial active immunity or stuff like that. So long story short, natural active immunity is for infection. Then you have artificial active infection, which is due to a vaccine. And then artificial passive is injection of immune serum. And then a natural passive is just antibodies passed from mother to child, such as IgG. All right, cool. Let's move on. These are just your 2 adaptive immune pathways, kind of like summarizing and bringing it back. So your humoral response to B cells and antibodies, and your cell-mediated response, which is your T helper cells and T cytotoxic cells and stuff like that. Next. All right, these are the phases of the humoral response again, the clonal expansion, the antibody secretion. These are just kind of like to help you really get into your head because a lot of it's memorization. All right, cool, next. And then a little bit about monoclonal antibodies to finish off. So this is basically identifying and immortalizing a single B cell that secretes an antibody. So monoclonal as in a single specific type of antibody. And what these are basically like drugs that they form to create the specific antibody to target a specific receptor or disease. So for example, they're really commonly used for autoimmune disorders and for cancer therapy. They'll, in your, I think your immunology tutorial that you guys are meant to have, you'll talk a bit about monoclonal antibodies and their role in cancer therapy, specifically in CTLA4. I didn't talk about it here, because I think you'll cover it later. And we're out of time anyways. All right, cool. Next slide. All right, so there are a bunch of practice questions that I've absolutely snagged. I won't run through them now because I'm already running way over time. If you guys have any questions, either message me or ask now before I hand over to Mahesh, who's hopefully here. Yep, Mahesh is here, and he'll be running through pharmacology. Okay, cool. Any questions? All right, cool.
Oh yeah, okay, I got one.
Yeah.
So during the development of the B and T cells in the bone marrow, when they're developing the tolerance, what happens if you have an infection?
So this is kind of like a continuous process. So there's not really like a point in your life where you won't have any B or T cells. if you know what I mean. So
Yes.
like this is like a continuous process of proliferation and maturing the T cells. And if you're considering maybe like when it's a brand new infant, it's still got like some immunoprotection from the mother through like IgA and IgG via the placenta. So it's okay. But like if there are issues with the bone marrow and bone marrow diseases, then it can affect the proliferation and growth of and development of these T cells and stuff like that. I hope that answers your question.
Yeah, thank you, thank you.
Okay, cool. Then I will get out of your hair. And Heshi, whenever you want to start, go ahead.
Am I
Thanks.
starting to
Thanks.
share?
Yes. Thank you so much, David.
No, it's all good. Thank
Absolute
you.
legend.
Thank you.
Sweet. Should we give them a 5-minute break, or shall we hop into it?
You can hop into it if you like. I think, well, it's your choice, really. Oh, well, I think this Zoom ends at 12:45, if I'm not wrong. So hopefully it's not too bad. I'm so sorry. I took a bit too much time, but hopefully not.
That's all right. Yeah, you had a lot to cover.
You guys don't worry too much. You'll be SWOT back revision lectures on both these topics as well. All right, cool.
Yeah,
Thanks,
what
Ashi.
I'll do, no worries. What I'll do is I'll give you guys a couple of minutes to just have a breather if you need to go to the bathroom or have a drink of water, and then I'll start. So for me, it's 11:25, so we can start at 11:28. Sounds good.
All right. If you have any questions or problems, Heshi, just message me on Messenger. I'll just leave the Zoom open to record, but I'll probably go do something else for a
Yeah,
while.
that's okay. Yeah, sounds good. Well, hello everyone. Let's get started. I'm Mahesh, your fellow second year, and I hope you're all well, happy, and healthy. Any other year would probably be in a couple of 100-seat lecture theatre, but nonetheless, we have to adapt. And here today on Zoom, I'll be taking you all through pharmacology. Before I do begin, I do want to thank Akil for putting this all together, and for that, the lecture on microbiology and immunology. I popped in for a little bit, and it was great to revise some of those concepts. So for those who haven't seen me before, this is me. This is also me, and this is me with facial hair. And essentially, my role here today is to try and live up to the legacy of this man right here, as well as Prof. Widdop and Bullock, the other farm lecturers. And I'll start with something that Richard always says, even though sometimes I don't like to admit it. That is, anatomy is cool. It really makes you feel like a med student. But when you're out there treating patients as doctors, you probably wouldn't go a day without administering some drugs, medications, or fluids. Hence, the vital importance of pharmacology. So with that, let's get right into it. So what we'll be covering today, I guess what I'll be doing is revising and going over the content that you guys went through for weeks 1 to 10 of pharmacology, and that includes these following topics. So some introductory terminology, pharmacokinetics, pharmacodynamics, the neuromuscular junction and the drugs that act there, as well as some anti-cancer drugs. Oh, just a note, if you guys have any questions along the way, just chuck it in the chat. I'll try my best to monitor it. If not, I'll get to it at the end. And stop me if you see any errors, because it's great to have a second pair of eyes. So, and just as important as what we will be covering is also knowing what we will not cover today. And that essentially is details of the nervous system, action potentials, and synaptic transmission. That's all covered in physiology. I won't be going through cancer, even though I'm going through anti-cancer drugs, because once again, that's not pharm and it's mostly covered in biochemistry. And I won't be going through antibiotics, antivirals, and antifungals. You guys will be going through that stuff over the next couple of weeks in weeks 11 and 12. And one of my peers will cover that content in the SWOT-VAC revision lecture. Okay, so the first section is looking at some introductory concepts. And I'll be breezing through this. I don't think you'll necessarily be assessed on this, but nevertheless, it is important to know. So we'll be starting off with some definitions and naming. So the definition of a drug is any chemical substance that brings about a change in biological function. And this does not include essential dietary nutrients. So it doesn't include water, vitamins, or minerals, for example. And they can either be endogenous. So this means it's produced naturally in the human body. For example, hormones. And it can also be synthetic. And this is essentially man-made chemicals that are usually produced in laboratories. And the idea of a drug is that they should work in small quantities. Now, when it comes to naming, we have three main ways we name drugs. The first name we give is called a chemical name, and it's essentially the substance is named based on its organic composition. And as you can see, the names can get quite long. So they're not really practical for a clinical setting. And that's why we have what's called a generic name, which is where the drug is named based on its mechanism of action, and it's given a drug class and a classification. And finally, we have something called a trade name, which is essentially when a pharmaceutical company has come along and branded a drug for commercial use. See, the examples I have here is salbutamol and Ventolin. Another example we can use to highlight this difference is, paracetamol would be a generic name. Many pharmaceutical companies manufacture it and distribute it. But a popular trade name that we might all be familiar with is Panadol, and that's produced by one specific company. There are a number of ways we can source drugs. So firstly, we can do it naturally through plants, animals, and microbes. We can do it through what's called serendipity. And this is essentially when a drug has already been developed to treat a potential disease or condition. But what we find is that it has a desired therapeutic effect for another disease. So we just essentially go take that drug and then start using it for this new disease or condition we have in front of us. We also can do it via drug design, which is essentially starting from scratch and designing a drug for a specific target. And finally, we can screen chemical libraries with already manufactured drugs to see if we can find one that suits what we're looking for. So this here depicts the three main phases of drug action. The first being the pharmaceutical phase. And this is essentially where the drug is quite literally, released from its package, released from the capsule you swallowed, being removed from a bag of fluids into the patient, whatever it is. Now, the next is the pharmacokinetic phase. And we like to think of this as what the body does to the drug. And finally, we have the pharmacodynamic phase, which is what the drug does to the body. And I I understand that you guys, the last thing you covered in pharmacology was pharmacokinetics, but that's actually one of the first things we're going to cover in the lecture today, just because it logically fits before, let's say, pharmacodynamics in the sequencing of drug action, even though pharmacodynamics is one of the first things you guys learned. So let's hop into pharmacokinetics. So there are 5 aspects of pharmacokinetics that we need to understand and cover. The first being administration. With administration, there are a number of definitions that we need to first understand when it comes to the dosage of drugs or whatever we are administering. And I think this diagram does a great job of summarizing a lot of these terms. Let's see if I can pop up this annotate tool. There we go. So I want to bring your attention to this first term called the minimum effective concentration. Actually, just look at the graph. So on the y-axis, we have an increased concentration of the drug in the plasma or the blood or the circulation. And on the x-axis, we have the time the drug has been in the patient after administration. So once again, let's go back to minimum effective concentration. And this is essentially the minimum concentration of the drug in the plasma that starts to produce a therapeutic effect in the patient. The next term is the maximum safe concentration, also known as the minimum toxic concentration. And essentially, this is the concentration in which a drug in the body will start to produce adverse effects. Now, the difference between these two concentrations is what's called the therapeutic range. So the therapeutic range is the range from the concentration in which we start to produce effects in the human body and the concentration in which we start to produce adverse effects, toxic effects in the human body. Now, I want to draw your attention to now onset, which is essentially the time it takes for the concentration of the drug to rise above the MEC. The time it takes for the drug to start producing an effect in the body. That's onset. And then the next, duration. This is the time, as we can see, that the drug concentration is above the MEC, the time that the drug is producing a therapeutic effect in the human body. Those are the terms that I want you to, that you guys need to know. And there is one more. So this slide essentially just spells out what I just described there on the diagram. So if you guys want to come back and have a look in case you've forgotten one, it's all here. But there's something that was not on the diagram, which is the therapeutic index. So what the therapeutic index is, it's essentially, it describes a relationship we see in the therapeutic range, but as a proportion. Now, if I go back, I should have mentioned this earlier. We want our therapeutic range to be as large as possible. The reason being is we want to produce an effect in the human body, the desired therapeutic effect, while minimizing the risk of potentially having too high of a concentration and producing adverse effects. Makes sense, right? We don't want to harm the patient. So going back to the therapeutic index, we also want this to be as large as possible. And it makes sense when we look at the individual components of the therapeutic index. So first, the MTC. Remember, we want the concentration of the minimum toxic concentration to be as high as possible, right? Because we don't want to reach that value. We don't want to produce adverse effects. And when this is high, because it's in the numerator, the therapeutic index will be high, which is what we want. Now, with the MEC, we want to try and produce remember we said that we want drugs to work in small doses. So we want this to be as small as possible. So when the denominator, the minimum effective concentration, becomes smaller, this will in turn become larger. So I guess the takeaway from the therapeutic index is the larger it is, the lower the risk we have at producing some sort of adverse effect in the patient, while at the same time producing the desired effect we want. Hopefully that makes sense. And with administration, we need to also be aware of the different routes we can take. You don't necessarily need to memorize all of these, but you do need to understand what they mean. So first, we can inject the drug into the patient. There are 3 forms. We've got intravascular, going straight into the veins and then into the circulation. We have intramuscular, which we do it into the muscle. And we also have subcutaneous, which goes into the fatty layer, just under our skin. And this relationship essentially demonstrates which form will have the most amount of drug going into the circulation. Because intravascular, by definition, you go straight into the veins, straight into the blood, we will get pretty much 100% of the drug going in there. Intramuscular, there are a bit more barriers because it has to go into the muscle and then sort of diffuse into the bloodstream. And then we have subcutaneous, which has additional barriers. But that just describes that relationship. The next is oral. I think that's self-explanatory. We have something called sublingual, which is where you put the drug under the tongue. And something to note about this enables the drug to go straight into the vena cava. And you guys will learn about this when you go to the cardio system. But essentially the vena cava is where all the veins in our body drain into before it goes into the heart. Intravaginal, we also have intraurethral. So the urethra is a tube that starts at the bladder. Inhalational, breathing it in. Intranasal, also breathing it in through the nose. Inhalational is through the mouth. And we also have transdermal, which is essentially anything topical on the skin, like creams and ointments. When it comes to administration, there are a number of factors that we have to consider about choosing which route. So our preferred route always is oral. The reason being the patient is more likely to go through the whole treatment regimen if they're taking oral medications. But there are times when we have to start considering, okay, do we need to start using some other routes? And there are some reasons why we would choose like an intravascular, for example, instead of an oral route. And these are some of the reasons. So the first being that the drug might be unstable or rapidly inactivated in the gastrointestinal system. So when you take a drug orally, it gets absorbed in the stomach. And if the drug gets inactivated in the stomach, for example, we probably don't want to use oral as a route. Also, when the absorption is compromised by vomiting or disease state, if the patient is unconscious, we can't give them oral medications. Maybe there are some systemic effects or adverse effects associated with taking the medication orally. So then we might consider another route as well. And also finally, there's this concept of bioavailability. I will be going through this a bit later, but it's the idea is, you know how there are more barriers in an intramuscular injection than an intravascular? They sound quite similar. IV versus IM, just like that, there are different barriers with oral compared to like other routes. Because oral, you've actually got to swallow drug, it's got to get absorbed in the stomach, and that has a low bioavailability. Essentially, less amount of drug will actually reach the circulation, where by definition, An intravascular injection, 100% of the drug will go in. But we'll be going through that very shortly. Yeah, so that's administration. The next idea is absorption. And we want to first look at, okay, how is the drug, how does the drug move in the body across membranes? So this is called permeation. And there's 4 main methods of permeation. The first being liquid diffusion. So this is essentially the idea of passive diffusion across membranes down the concentration gradient. The second method is aqueous diffusion, and this is via protein channels, via aqueous channels. The third is carrier-mediated transport. This is active, if you remember, active and facilitated diffusion across the membrane. And finally, we have exos or endocytosis for large molecules and bulk movement. Now, there are a couple of main factors that affect the movement of drugs across membranes when the drugs are being absorbed either into circulation or tissues. And the first being ionization, which I'll be going through on the next slide. And the second being its molecular shape and size. And the idea is the larger the shape the molecule is, the more difficult it is for it to move across the membrane. So the larger it is, we have a lower permeation. And just a note, ionization affects permeation more than size. So let's have a look at ionization. Now, essentially what ionization is the drug's charge. It's the state of the drug, whether it's charged or uncharged. So for example here, less ionized means the drug is less charged, and the less charged the drug is, the more lipophilic it is. Remembering that the surface of cell membranes are lipophilic, so that favors the drug passing through the membrane. So yeah, so the more lipophilic it is, the better permeation of the drug across the membrane. And you may come across the term ion trapping in lectures or in notes or in textbooks. And what ion trapping is the opposite of this process or this equation that we just went through. Ion trapping is, the more ionized the drug is, the more trapped it is, the more unable it is to pass the membrane because it's less lipophilic and it can't pass through. And why ionization is important is because drugs change their ionization state based on the pH of the environment, or how acidic or how basic the environment is. And as we know in the body, the pH varies in our body, and depending on where the drug is in the body, whether it be in the stomach, which is a quite acidic environment, or in the small intestine, which is quite a basic environment with a high pH, you want to know, okay, is the drug going to be ionized, unionized? How is that going to affect the absorption at those different levels in the body? Just before we hop in to explaining this, just want to revise a bit of chem, which is remembering that acids donate protons and bases accept protons. So this whole idea of, okay, that's important to note because some drugs will tend to be more acidic and some drugs will tend to be more basic. And depending on that, we will see how it becomes ionized or unionized. And that's sort of described by this equation here. And I'm going to try and break down this equation for you, and hopefully that will cover how ionization changes with pH. So at the top, we have drug B, which is a basic drug. It likes to accept protons. Now let's have a look what happens to a basic drug in an acidic environment, let's say the stomach. Actually, let me take that back. So B on its own is an unionized form of the drug. It's not charged. And on the left-hand side of the equation, it's in its ionized form, it's charged. Now let's have a look at what happens in an acidic environment. So the way I want you to think about is what's happening to the H+, remembering the H+ is what determines the pH. So the higher the amount of hydrogen protons, the lower the pH, the more acidic it is. And the lower the amount of H protons, the higher the pH and the more basic it is. So let's have a look at what happens to the H+ here. So in an acidic environment, we'll have high amounts of this proton. And if we remember the concept of equilibrium from chemistry, the high amount of this proton, the more of this there is, the more this equation is going to be driven to the left. So in a more acidic environment, this equation will be driven to the left-hand side and the drug will become more ionized. Now let's have a look at what happens in a basic environment. In a basic environment, we'll have less protons. The equation will be driven to the right. So the drug will favor being in an unionized state. So essentially, basic drugs tend to be more ionized in acidic environments and more unionized in basic environments. Now let's have a look at acidic drugs, where the acidic drug here is drug A. Here it's unionized, and here it's ionized. So what happens in a basic environment? Let's start with that. There's less protons, so then the equation will be driven to the right, and it'll be in an ionized state. Now, in an acidic environment, we have more protons here. The equation will be driven to the left, and it'll be more unionized. So in summary, basic drugs like to be in basic drugs will be absorbed better in basic environments because there's less protons and the drug will favor being in an unionized state, which is good. It's more lipophilic. It can pass through membranes. And acidic drugs are better absorbed in acidic environments because we have more protons here. The equation is driven to the left, being in an unionized state, so then it can be absorbed and pass through membranes. If you'd like me to go, if anything doesn't make sense, once again, just chuck in the chat. And then this is where this term comes along. There's this term called pKa, and all the pKa is the pH where the drug is 50% unionized and 50% ionized. So if you have a pKa of 10, at the pH of 10, that drug will be 50% unionized and 50% ionized. If the pKa of a drug is 7 or above, it tends to be more of a basic drug. And if the pKa is 7 or below, the drug tends to be more acidic. Now, yeah, that's all. So just, this just sort of summarizes what I was discussing before. So drugs with a high pKa, basic drugs, they're absorbed better in a basic environment and poorly in an acidic. And drugs with a low pKa, they're more acidic drugs, and they're absorbed better in an acidic environment and poorly in a basic. And just sort of applying the concept that we learned from here with these equations. Let's say we take example a drug of atropine. It has a pK of 10, so it's more of a basic drug. If the pH is 11, so it's more basic, we will have more unionized form of the drug than ionized. We'll have more than 50%, and we have less than 50% ionized. But if atropine is in an environment of a pH of, let's say, 9 or 8, that's more acidic than its pKa, so it doesn't like that. So more of the drug is going to be ionized, and less of the drug will be unionized. So essentially pKa, we have sort of some balance 50-50, and depending on whether it's more basic or more acidic, these percentages or proportions will change. Cool, that's ionization. Okay, so I mentioned a bioavailability before. So that relates to the absorption of the drug. And essentially the definition is bioavailability is a measure of how much a drug is absorbed into the systemic circulation after oral administration compared with IV, but we can also compare it with other forms. So by definition, as I said before, intravascular injections have 100% viability because all the drug is going straight into the blood. But then oral will not ever reach this number because of these factors. So there's absorption time across the gut. There's something called first pass metabolism of the liver. And essentially, once the drug is absorbed across the gut, the liver will have a look at that drug essentially before it goes into the blood, and will actually remove some of it. And also its pharmaceutical formulation, like how it's created, will affect it. This little graph in the bottom right-hand corner sort of highlights the differences. So intravascular, we can see it starts off with 100% bioavailability, and then as it's metabolized, as it's excreted, it decreases over time. The next being intramuscular, then subcutaneous, and then finally oral. So that's absorption. The next thing we got to look at is distribution. So the way I like to think about distribution is how much of the drug is being delivered to the tissues. There are a number of factors that affect this. The first being, the movement across membranes. If the drug can't move across the membrane into the tissue, it can't go there. And a small note is that only small fat-soluble drugs can cross what's called the blood-brain barrier, meaning that only small fat-soluble drugs can actually get into the brain. Blood flow affects distribution because obviously the more the blood is flowing, the faster the blood is flowing, the quicker the drug can get around to the different tissues in the body. Also, how efficient the drug is at binding to tissues will affect how quickly it would be absorbed. It'll actually be delivered to tissues, and also binding to plasma proteins. So if the drug likes to bind to plasma proteins in the blood, it's more likely to stay in the blood and less likely to go and bind to tissues and be delivered to them. And just an example here, there's a protein, plasma protein called albumin. When the drugs are bound to albumin, we like to describe it as albumin, this protein reduces the amount of free drug, which can then go and act at tissues and produce some sort of therapeutic effect. And a measure that we use to sort of quantify distribution is what's called volume of distribution. And essentially what this does, it compares the dose, or I guess the total amount of the drug in the body, to the concentration of that drug in the plasma, in the blood, in the circulation. And essentially, the idea is this sort of relationship is determined by sort of the binding strength of that drug to either tissues or plasma proteins. If the drug is really good at binding to tissues, less of that drug will be in the plasma compared to the amount of drug in the body, which will sort of make the volume of distribution higher. Now, if the drug likes binding to plasma proteins and staying in the blood, this will be more similar to the total amount of drug in the body, because there'll only be a lower amount in the tissues. So the volume of distribution will be closer to 1. And this means that essentially we know that the drug must be quite good at binding to plasma proteins because it's staying in the blood. So that's what I've got here. So a high volume of distribution high dose compared to the amount in the plasma means that they like to stay in the tissues, and that's sort of the term used for that. And if the dose is similar to the plasma concentration, then we know the drug is staying in the plasma in the blood, so it must tightly bind to plasma proteins. What we've got here is metabolism. And there's this term we have when we used to, it's essentially another word for metabolism, but related to pharmacokinetics, and it's called biotransformation. And all this is the chemical alteration of the drug. So biotransformation mainly occurs in the liver. It can also occur in the gut, in the plasma, and in the kidneys. And there's this group of enzymes that undergo will help assist with the process of biotransformation. And these group of enzymes are called CYP450. And this is essentially the main group of enzymes that metabolize drugs, that alter drugs chemically. And what these enzymes do, they make lipid-soluble drugs more water-soluble. And this is important because kidneys like excreting water-soluble drugs. When they're lipid-soluble, it's really tough for the kidneys to excrete them. And there's two sort of relationships that we need to, I guess, conceptualize when it comes to metabolism, sorry. And they come under what's called dose-dependent kinetics. So the first relationship is first order kinetics. And what this means is that the rate the drug is being metabolized in the body is proportional to or is dependent on the concentration of the drug. First order follows the relationship that we've all understood with enzymes and solute relationships. So the higher the concentration of the solute, the faster the rate of reaction, because more solutes are sort of colliding with the enzymes and reacting. And first order kinetics happen when there is enough enzymes to metabolize whatever drugs you're consuming in the body. Now, the second relationship, which is we don't see it as often, but very important to note, is zero order kinetics. And this is where the rate of the drug metabolizing the body, or rate of reaction, stays constant, and it does not change depending on the concentration of the drug. And this happens when there's not enough enzymes. So the enzymes are always saturated, so they can only sort of metabolize a limited amount of drugs per unit of time. And a great sort of example to think about this is, alcohol. Whether you drink 5 standard drinks or 10 standard drinks, you will always, continue to sort of eliminate that from your blood at a constant rate of 1 standard per hour, whatever it is for you. Where, if it followed first order kinetics, if you had, 10 versus 5, maybe when at 10, you might remove 2 or 3 rather than at 5 at 1, but it doesn't work like that. So that's the ideal. And what we need to notice is, as the concentration of the drug increases, our enzymes in our body, particularly these CYP450s, will become more saturated, and we will move closer and closer towards zero order kinetics away from first order. And just as I said before, some examples of zero order kinetics, we have alcohol, but we also have, and this is at any concentration, and we also have aspirin and phenytoin in medium to higher doses. And we need to always keep note of zero order kinetic drugs because there's always a really good risk of overdose. And the final sort of concept with pharmacokinetics is elimination. There's a number of ways we can excrete the drug in our body. as I said before, firstly with the kidneys, which like to excrete water-soluble drugs. We also have the gut, which excrete via the bile and feces. Lungs excrete gases and volatile liquids. And we can also excrete drugs via sweat, saliva, mucus, tears, and milk. And just a note, we don't want to give drugs that pass through the milk to lactating women. They like to ask questions about that, so just keep that in mind. Okay, we've had a look at what the drug does to the body. Now let's have a look at what the body does to the drug. Now let's have a look at what the drug does to the body. So the first thing we need to look at are the targets for the drug in the body at a more of a molecular level. So the targets of drug action include ion channels, those aqueous channels and that we were talking about before, enzymes, transporters, so these, those carrier proteins, and finally receptors. And receptors are quite important. It's one of the most predominant things that we really need to take note of when it comes to administering medications. And that's why I want to look into it a little bit more detail. So what receptors are? They're proteins either found on cell membranes or intracellularly. So this sort of diagram helps depict that. So we can have a receptor on the cell membrane, or we can have a receptor intracellularly. And essentially these receptors bind to ligands, which in this case are the drugs, via specific recognition sites. And upon binding to that ligand, that drug, it will transmit a biological signal to produce some sort of an effect. And there are two terms we've got to understand when it comes to drug receptor interactions, and that is selectivity. And it's the idea that the drug will act on wherever the receptor exists. So no matter where the receptor is in the body, the drug can bind to it and it can act on it, whether that be in the heart, the lungs, the blood vessels. And then there's something called specificity. Love this word, specificity. It's where the drug will act on its receptor at a specific organ. So the drug has been designed to sort of target its specific receptor on one organ only, so only the heart, even if the receptor is found on the lungs and so forth. But in general, with how we manufacture drugs, most drugs are selective, meaning that they will act on that receptor that they're designed for, no matter where the receptor is in the body. There are a number of ways we can measure drug binding interactions, actually 3 ways here. The first being affinity. And what this measures is the strength of the drug binding to a receptor. Is that strength high or low? So you can have low or high affinity. And a measure we use for this is Ka, and that measures essentially the affinity of binding, and it's a positive relationship. So the higher the Ka, the higher the affinity, the higher the strength of the binding. We also have efficacy, which is the capacity for once the drug has bound to the receptor, for that complex to generate some sort of response. E, this funny E, sort of represents efficacy, and it's a positive relationship once again. And a term that you might come across quite a bit when it comes to efficacy is receptor reserve. And what this is, it's essentially where more receptors are available than required for a maximal response. This example sort of explains it. So if only 5%, if a drug only requires to bind to 5% of the receptors in the human body to produce its maximal response, the receptor reserve, the receptors that they don't need, is 95%. And this diagram here sort of depicts affinity and efficacy well. So affinity is the strength of the drug binding to the receptor, and that's measured by Ka. So once it's bound, its ability to produce some sort of response is efficacy. And finally, we have something called potency, which is the drug concentration, which produces 50% of a maximal response. The measure we use is EC50, and the difference with this is it's an inverse relationship, meaning that the lower the EC50 we see, the higher the potency, the higher the drug concentration we need to produce 50% of the maximum response in the body. We also have to know the different types of drug binding interactions. So there are a few terms that we need to know. So the first is agonist. This is where the drug binds to the receptor and it activates it and it produces some sort of effect. It has affinity and it has efficacy. A partial agonist, on the other hand, is it binds to the receptor, but it produces a lower effect. So it may have the same affinity, but a lower efficacy. So a way of saying this is it may occupy the same amount of binding sites, but it will produce a lower effect in the body. We also have an antagonist. It's essentially where a drug binds to a receptor, but there's no effect. It cannot activate it. So it has affinity, but has no efficacy because there's no response. And there's a few types. So there's competitive reversible, which means although it may bind to the receptor, if we increase the concentration of the agonist, it can compete for it and bind instead. We also have competitive irreversible, which stays stuck to the receptor, and non-competitive, different receptors for antagonists and agonists. And then a little funny one is inverse agonist. This is where the drug binds and causes sort of essentially the opposite effect of an agonist. So it has affinity, it has efficacy, but its efficacy is in the opposite direction. And I think this diagram is one of the best out there when it comes to explaining the difference. So we have our full agonist, it binds and it produces a strong effect. A partial only produces half the effect. An antagonist, remember, it's neutral because it binds but has no efficacy. And an inverse agonist binds and it has sort of an opposite effect. And if you still don't quite get it, there's this key and lock analogy that helps you understand it. And where the key is the drug, the lock being the enzyme or the receptor, where with an agonist, the key fits the lock, it has affinity and it opens it, it has efficacy. An antagonist, it fits the lock but doesn't open it, it has no efficacy. A partial agonist is where the key fits the lock and sort of half unlocks it. And this is where the analogy comes somewhat a bit of a stretch, but an inverse agonist is where the key fits the lock and somehow makes it more locked. Maybe the key gets stuck in the lock and it's harder to open now. Whatever you need to make the analogy work. Cool, that's covering drug action more from a theoretical level. Now we're going to have a look at the neuromuscular junction and the drugs that work there. So here's just a bit of a revision of the divisions of the nervous system, not quite pharmacology, but I think it's useful to have a look at. So the nervous system, it's divided into our central and our peripheral. Central nervous system comprises of the brain and spinal cord, which is depicted in yellow on this diagram. And the peripheral nervous system, we have what's motor neurons. These sort of carry out effects. It stimulates muscle, and sensory neurons where we sort of respond to stimuli in the environment. Once again, sorry, the PNS here, it's the green. And the motor neurons are split into somatic and autonomic. Somatic here is voluntary, it controls voluntary muscle movements. And autonomic sort of happens automatically without us thinking about it. It's involuntary muscle movements. And with voluntary muscle movements, that's with what's called skeletal muscle. So if we want to grasp our hand, we're using skeletal muscle here. And stuff that's happening automatically, that's with smooth muscle. So, our heart's beating without us knowing, and it's comprised of smooth muscle. And within the autonomic nervous system, we have the sympathetic and parasympathetic systems, where the sympathetic is sort of known for its fight or flight-like responses, and the parasympathetic for its rest and digest. So this sort of favors us digesting food and sort of not running for our lives, and this favors us, fighting and increasing our heart rate so we can run fast, etc. And today we're focusing on the neuromuscular junctions of the motor neurons in the peripheral nervous system, our voluntary movements and our involuntary movements. So I'm going to spend a little bit of time sort of breaking down this image because it quite well summarizes all the neurotransmitters we need to know and all the receptors for the 3 systems we're looking at in the PNS. Just to acknowledge something, it's not the focus for today, but our autonomic nervous system, our parasympathetics and sympathetics, our whole nerve is split into 2 neurons, where our voluntary movements with the somatic, it's just one nerve straight to the neuromuscular junction. That's just something to acknowledge, but our focus for today is at this neuromuscular junction, knowing the neurotransmitters and knowing these receptors really well. So the first thing is knowing these neurotransmitters. So ACH is acetylcholine, NA is noradrenaline, but we can also have adrenaline here. And acetylcholine works on cholinergic receptors. Sort of makes sense because it has parts of it in each name. So acetylcholine, cholinergic, acetylcholine, cholinergic, and noradrenaline acts on adrenergic receptors, noradrenaline, adrenergic receptors. Now there's 2 types of cholinergic receptors. There's a muscarinic. When I think of muscarinic, I'm like, okay, the opposite of us being able to control our muscles. So, which is weird. And that's linked with the parasympathetic nervous system because this is involuntary. And then we also have nicotinic, which is associated with our somatic, our voluntary muscle movements, and that's that deals with skeletal muscle. And then there's also 2 subtypes for our adrenergic receptors, our alpha and beta. You do need to remember which is found on what, but we'll deep dive on that a bit later. So once again, yeah, acetylcholine, remember cholinergic, noradrenaline acts on adrenergic. Remember whenever you see muscarinic, think, it's the opposite. We're not controlling skeletal muscle. So parasympathetic system, and then nicotinic being the somatic system. And of course, we have our 2 subtypes, alpha and beta here. So just a deep dive at what happens at one neuromuscular junction. What is it? I mean, neuro meaning nerve, muscular meaning muscle. So it's the junction, it's the interaction between a nerve and some sort of muscle, whether it be smooth or skeletal. So what happens here, the nerve impulse comes, the action potential, it comes into the axon terminal of the nerve. This stimulates voltage-gated calcium channels to open, allowing an influx of calcium. This influx of calcium sort of stimulates snare proteins to attach to these vesicles that contain our neurotransmitters. These neurotransmitters have been synthesized prior and have been stored in these vesicles. The snare protein takes it to the presynaptic membrane, where then the neurotransmitters undergo exocytosis into this space called the synapse. The neurotransmitters will diffuse across, bind to some sort of receptor, nicotine, it could be a cholinergic, it could be an adrenergic receptor on our postsynaptic membrane. This triggers an action potential, which then travels down what's called a transverse tubule, which then essentially in turn, there's this DHP protein that sort of senses it, which would then, which transmits the signal to the sarcoplasmic reticulum. And then the sarcoplasmic reticulum will release calcium via what's called a ryanodine receptor into the cytoplasm. And then these calcium ions will go and stimulate a muscle contraction. So to summarize, action potential comes into this terminal. Neurotransmitters exocytose, they bind, cause an action potential in the muscle. Calcium is released from the sarcoplasmic reticulum to cause muscle contraction. How that calcium does it, that's a completely different process. That's something you don't need to worry for now. Something that we do need to note though is what that's important in terms of pharmacology is what happens to these neurotransmitters after this process at the synapse. One of three things happens. So they can either be taken back into the presynaptic neuron and recycled via reuptake. This is called reuptake. They can either be they can also be broken down by enzymes that sort of lie in this synapse. We got an example here of of a cholinesterase, or they can just sort of just diffuse out, and this will stop the stimulation here. So once again, I just spelled out what happens there in words here, if you want to have a look at it again. And now let's have a look back at, okay, we know what happens at one junction. What are the specific effects on the organs at these different types of, with the different types of systems? Once again, remembering, I'm going to keep emphasizing it: acetylcholine, cholinergic, noradrenaline, adrenergic, and then remembering the subtypes. It's muscarinic, it's not voluntary muscle movements, so it must be the parasympathetic, and then nicotinic is with the somatic. I'm going to be keeping that diagram in the top right, because I know it can take some time to just sink in. I know for me it did. So I'm going to leave it up here so you can always keep referring to it and seeing what we're looking at when we dive into the, I guess, the different effects with the different nerves. So first thing we're looking at is the adrenergic nerves. Adrenergic is part of the sympathetic nervous system, which will produce effects that help us in fight or flight situations. The neurotransmitters, once again, are noradrenaline and adrenaline. And this table summarizes the effects we'll see at when the adrenergic receptors are activated. You do need to remember the subtypes, but I always want you to think when we sort of guessing what effects we might have, go back to the idea of fight or flight. So in our gut, which is responsible for digestion, we kind of don't want that to happen when we're running for our life. We don't want to rest and digest. So we'll have, if one of these are stimulated, we'll have a decrease in motility for the heart. We have beta subtype. We will have an increase in contraction because we want more blood to go to our tissues. With our lungs, we want to breathe more oxygen. So we'll have bronchodilation, opening up our airways. And with our blood vessels, we'll have vasoconstriction, which increases our blood pressure and increases the speed in which the blood travels around our body. We also need to know some of the ways the neurotransmitters are modulated at this sort of junction. So when some amphetamines, which is a sort of a recreational drug, they increase the release of noradrenaline, which will increase stimulation of the receptor, increasing these effects we see here. These MAO and COMT molecules, they help with the process of noradrenaline reuptake into the presynaptic neuron. These are their names, if you're interested. And cocaine inhibits COMT, So it inhibits reuptake, so there'll be more noradrenaline at the synapse, meaning there'll be more stimulation and more of these effects. And at the response, meaning at the postsynaptic membrane with the receptors, agonists and antagonists can also sort of change what type of response we might see. Next thing we need to look at, we did adrenergic. Now let's look at cholinergic. So cholinergic, we have our parasympathetic nervous system. Remember, thinking of effects in relation to rest and digest. We have our somatic nervous system, which is responsible for voluntary muscle contraction. Parasympathetic, once again, we're thinking muscarinic subtype and nicotinic subtype here. And our only neurotransmitter we need to worry about is acetylcholine. So here are the effects in a table. So muscarinic receptors, we're looking at the parasympathetic nervous system, looking at rest and digest. So at the heart, we're chilling out, we'll have less of a contraction. With our blood vessels, we don't need fast blood flow, so we'll have vasodilation, decreasing the blood pressure. And we do want to digest now. We're good to rest and digest, so we'll have increased motility of the gastrointestinal system or our gut. Now, our nicotinic receptors only work on skeletal muscle, and when they're stimulated, we'll just have contraction of that muscle. Okay, how are neurotransmitters modulated? So at these junctions, these cholinergic neuromuscular junctions, well, there are these enzymes called cholinesterases, and these break down acetylcholine. So there's a substance called anticholinesterases, and they sort of act as antagonists and bind to cholinesterases, and these stop these enzymes from breaking down acetylcholine, so more of it will be stimulating our cholinergic receptor, meaning it could lead to overstimulation. And this overstimulation can actually cause what's called anticholinesterase poisoning, and we need to know some of the symptoms of that. And the next slide will sort of give us some mnemonics and some ideas so we can remember what happens when our muscarinic receptors are overstimulated and our nicotinic receptors are. So here are the symptoms for anticholinesterase poisoning. So muscarinic being our parasympathetic nervous system, we have our mnemonic of sludge, which stands for salivation, lacrimation, which is tears, urination, defecation, GI cramping, because we're overworking our gastrointestinal system, and emesis, which is vomiting. For our nicotinic, remembering this is our skeletal muscle, our voluntary muscle movements, we have Monday, Tuesday, Wednesday, Thursday, Friday. M standing for muscle cramps. We're overly contracting our skeletal muscle. We can also get tachycardia, muscle weakness, twitching, and fasciculations, which is also another form of muscle twitching. And finally, here are the neuromuscular junction drugs. There are only 4 you need to know on the ePharmacology core drug list, and they're here. And using what we've just learned about, the effects that adrenergic and, this actually only works on cholinergic nerves, but just using those, I guess, concepts, we can sort of figure out what happens. So the first drug is rocuronium. Its class is a non-depolarizing blocker. It essentially relaxes muscle. And it's a competitive antagonist at nicotinic receptors. Remember, this acts at skeletal muscle. And when skeletal muscle, if the skeletal muscle cannot contract, it stays relaxed. And it's essentially called like relaxed paralysis. And if there's too much stimulation over a long period of time, we can have the adverse effect of prolonged paralysis of skeletal muscle. And it's competitive, so it can be overcome by increasing acetylcholine, which can then compete for those nicotinic receptors. Our next drug is succinylmethonium, which is a depolarizing blocker, which also muscle relaxes. It's an irreversible agonist. So what at nicotinic receptors, once again, skeletal muscle, and essentially what happens is it will bind to the nicotinic receptor, causing an initial contraction, but because it does not leave the receptor, it cannot be stimulated again. because how muscle contraction works, it has to be constantly sort of stimulated, which is sort of, you'll learn in physiology. So it prevents like repolarization and for the muscle to contract again. So it causes paralysis as well. And here are some side effects. Okay, cool. And then our next drug is pyridostigmine. It's a reversible anticholinesterase, so it stops the breakdown of acetylcholine, and this sort of allows further activation of our muscarinic and nicotinic receptors. And our adverse effects, remembering overstimulation of these receptors lead to sludge and days of the week symptoms. And it's indicated in myasthenia gravis, which I'll be going through in the next slide. And we also have atropine, which is an anticholinergic, acts at cholinergic nerves, but only at muscarinic receptors, only in your parasympathetic nervous system. It's a reversible competitive antagonist at these muscarinic receptors, and the adverse effects are the opposite of sludge because we're having understimulation here, because it's sort of just sticking there at the muscarinic receptors and not, I guess, being stimulated anymore. And this is also indicated in myasthenia gravis. So what is myasthenia gravis? Why we use these two drugs? We're looking at specifically the somatic nervous system, our voluntary muscle movements with skeletal muscle. It's an autoimmune disease where our own antibodies attack our nicotinic cholinergic receptors. So there's less of these receptors to be sort of activated by acetylcholine. And essentially increasing muscle weakness, one of the symptoms we'll experience is increasing muscle weakness after activity, because our body's having difficulty contracting that muscle because there's less nicotinic cholinergic receptors. But we do recover after a short period of rest because this sort of comes back to a balance, this junction. So how do we treat this condition? We use pyridostigmine. Remember, we were stopping the breakdown of acetylcholine at this junction, so we can increase the stimulation here. We have more to stimulate these receptors, so that helps. And we also, but what we find is pyridostigmine, remember, works at both nicotinic and muscarinic receptors. So we're starting to have an overstimulation of the parasympathetic nervous system when we only really want to work at the somatic. So that's why we have atropine, which is an anticholinergic specific to muscarinic receptors, and we can block those sludge symptoms. And we also have corticosteroids or immunosuppressants, so we can suppress sort of the immune response in autoimmune disease. And that is NMJ. We're up to our last bit. I believe I only have 10 minutes, but we have time to get through this. Essentially, there's three main categories of anti-cancer drugs that we have to get through. Classic cytotoxic, there are 4 drugs for this. Hormones and hormone antagonists, there are 2 drugs for this. And novel therapy, which there is 1. Oh yeah, and I just want to have a look at this first before we go in, because we know this is the real reason why you turned up for the farm lecture today, which is knowing how to do drugs properly. I mean, this is the real reason that med students like pharmacology, for real. We all know what we do in our recreational time. So I'm going to spend some time going through this, and then we'll hop into anti-cancer. No, I'm just kidding. So what I mean by how to do drugs is how do we learn them? This is sort of the approach and mindset we need to take, because otherwise you're just going to be sitting there trying to learn drugs, and it's really not that helpful. The first point we need to know is only learn the ePharm core drugs. I know there's a lot of drugs that lecturers refer to in lectures, but they're sort of used to as examples to highlight concepts, and they're not necessarily drugs you need to know. So refer to this list to just know the drugs that you need to know. And all the drugs on this sort of presentation, they're all core drugs. You must know these. You don't need to know any others. And how do we and this is sort of the priority of what we need to know for the drugs in order. So prioritize knowing the mechanism of actions of all the drugs first, then the indications and contraindications. So indications being like what do we use it for, contra meaning what we don't use the drug in because there may be some adverse effects, like such as pregnancy, for example. And there's also next being mode of administration for antibiotics, and finally significant side effects. And some tools we can use to help us with this process are mnemonics, deriving information from the name of the drug, and analogies, which you'll see in some of the examples that I'm going to go through. Okay, so the first category is classic cytotoxic drugs. Essentially, these block protein growth in cancer cells, so then the cells can't grow. So the production of protein. The first drug is cyclophosphamide. Let me say that again. Cyclophosphamide. It's classed as an alkylating agent. So the mode of action is it inserts an alkyl group into DNA. This alkyl group causes cross-linking between strands, which is essentially covalent bonds, particularly between guanine bases. This cross-linking causes stress, the strand will break, and this causes apoptosis of the cancer cell. And some side effects are this drug targets rapidly proliferating cells, so not only cancer cells, but also things like hair, our sex cells, our cells in the bone marrow, so we can get hair loss, infertility, and bone marrow suppression. Now, using the actual drug to help us remember what it does, cyclo, we can think cyclone. Cyclone is fast, we target rapidly replicating cells. And fam, the guanines get together to make a family, and this is essentially referring to our cross-linking here. Next is doxorubicin. It's a cytotoxic antibiotic. Essentially inhibits transcription and translation in cancer cells. This impairs growth of it, and this allows the immune system to sort of start compensating, starting to take down the cancer. It's a cytotoxic, it's an antibiotic, but it's too toxic to be used in infectious disease. Our side effects are essentially the same, but with some gastrointestinal disturbances like vomiting or diarrhea. And from the name, dox sounds like docs, doctors repair. So the drugs stop the repair of DNA, inhibits transcription and translation. Next, we have 5-fluorouracil. It's an anti-metabolite. So what these do is they substitute themselves in the DNA synthesis pathway and blocks it, makes the DNA non-functional. This mode of action is it's a pyrimidine antagonist, so pyrimidine bases. So we're looking at cytosine, thymine, uracil, and it's S-phase specific in the cell cycle. Side effects, the same thing. And then tools, 5 kind of looks like an S, so S-phase specific. Uracil reminds us that it's a pyrimidine antagonist. And if you can cut yourself on a pyramid, so that reminds us of the bases, CUT. Our final cytotoxic drug is methotrexate, which is also an anti-metabolite, but this time it's a folate analog. Essentially, methotrexate inhibits this enzyme. But just remember, it's a folate analog. And our first sort of indication to remember is it's used in rheumatoid arthritis, and we can't use it in pregnancy. Same side effects. And how do we remember it? Well, you wouldn't give a pregnant woman meth, so don't give them methotrexate. That's a contraindication. And 8, folate, the folate pathway is inhibited. Now, we got 2 hormone and hormone antagonists. Why is this important? Well, sex hormones are required as growth factors for many reproductive cancers. So that's why we have these drugs, especially breast cancers. So our first drug is tamoxifen. It's classed as a selective estrogen receptor modulator, a SERM. Just got to remember that. Its mode of action is it's an estrogen antagonist. This is a hormone, female hormone, in breast tissue receptors. That means it's indicated in estrogen-dependent breast cancer. Our side effects are menopausal symptoms, hot flushes and sweating. And how to remember it? Well, Tam, well, she sounds like Tammy, which is a girl, so therefore it's a SERM, and she also has breast cancer. Sad face. Our next drug is anastrozole, which is an aromatase inhibitor. So it inhibits the formation of estrogen because aromatase converts this other hormone, androgens, into estrogen, and it's indicated in estrogen-dependent breast cancer in post-menopausal women, so older women. Same side effects, but with some GIT disturbances as well. And how we remember it? Well, Anne, we're thinking about Anne, who is that old lady down the street that smells funny. I don't know why it sounds like that. I deeply apologize for any Annes that may be listening. It's not too late to change your name. No, I'm kidding. But this has just been passed down through med students for a while. And our final drug is a novel anti-cancer drug. Novel anti-cancer drugs are sort of the new emerging fancy treatments. And we have trastuzumab, MAB meaning monoclonal antibodies, which is the class. And what these are, they they bind to and block HER2 receptors. HER2, that's what it stands for if you're interested, which is overexpressed in some breast cancers. So blocking that, we stop proliferation. It's indicated in HER2 positive breast cancer. Our side effects are cardiac dysfunction and gastrointestinal disturbances. And how do we remember it? Two girls wear tutus. So yeah, HER2 receptor inhibitors, they're her receptors, not his receptors. And yeah, good on you guys for staying to the end. I mean, look, you guys had micro and immunology just before this, and then you stayed for pharm. So good on you for staying through it all. This is just a summary of what we went through. Key takeaways, make sure you understand the concepts. I don't care if you remember the details, but I think this presentation was mostly looking at conceptual information. Make sure you do drugs the right way. And just a note that this is all high yield. I would say that if you're not sure what to revise for weeks 1 to 10, probably just use this presentation. It'll do you good. And yeah, that's all from me. I mean, I don't know if there's going to be time, but if there's any questions, I think there is. But I'll give I guess a minute for questions to come through. But where to find me, you can email me, you can find me on Facebook, you can find me on Messenger. And I guess as a parting gift, I also got a bunch of questions that have been directly sourced from past exams that sort of relate to this content, which are so the rest of the slides, and you'll find them in the post slides. I think Akil will, I'm going to give that to Akil, and he'll sort of give it to everyone else.
Yeah, guys, so sorry for having to close it off. The recording's just about to end, but thanks so much for coming. Thanks again, Heshi. Great presentation. See you guys later.